Good morning students. Already in the previous video, we have seen the structure of DNA and DNA replication. Replication means making exact copies of its own structure. In DNA replication, how many enzymes are involved? Yes, 5. What are they? Enzyme helicase, topoisomerase, primase, polymerase and ligase. What's the use of DNA ligase? It is used for joining the fragments. And today in this video, we are going to learn mutation. Mutation means sudden change in the genetic material of an individual. So genetic material, what do you mean by genetic material? Yes, DNA is the genetic material. So DNA, deoxyribonucleic acid. DNA is the genetic material. So define mutation. Mutation means sudden change in the genetic material of an individual. The term DNA, it was first introduced by Hugo de Vries. The term mutation was first introduced by Hugo de Vries in the year 1901 when he observed the phenotypic changes in the evening primrose Aneuploidy lamarckiana. So the evening primrose is Aneuploidy lamarckiana. So the evening primrose is Aneuploidy lamarckiana. So what's the botanical name for evening primrose? Aneuploidy lamarckiana. Here O silent. Okay. So the botanical name for evening primrose is Aneuploidy lamarckiana. And this mutation, it's of two types. They are chromosomal mutation and gene or point mutation. So they are chromosomal mutation. Chromosomal mutation and gene or point mutation. So first coming to chromosomal mutation. So from the name what we are coming to know? Yes, sudden change occurs in the chromosome. So sudden change which occurs in the structure and number of chromosome is called chromosomal mutation. Structural chromosomes usually occurs due to errors in cell division. So structural chromosomes, they usually occurs due to errors in cell division. Changes in the number and arrangement of gene takes place as a result of addition, deletion, inversion, duplication in chromosome. So addition, when you are taking the normal chromosome, you see the normal chromosome is like this. So this is the normal chromosome. So this is the normal chromosome. So see here, if this A is added, so here if this A is added, that is said to be addition. Okay, so if this A is added, that is said to be addition. Next. So here if A is added, that is said to be addition. And here you see if this A is deleted, that is said to be deletion. If this A is deleted, that is said to be deletion. And here you see if the C is coming here and if this D is going here, then it is said to be inversion. It is said to be inversion. So this is the normal chromosome. Changes in the number and arrangement of genes takes place as a result of addition, deletion, inversion, duplication. So this is the normal chromosome, and here if an A is added, that is said to be addition. And here you see this A is deleted, so that is said to be deletion. And here you see they are interchanged, so that is said to be inversion. Sometimes set of chromosomes will be added or set of chromosomes will be deleted. That is said to be ploidy. So sometimes set of chromosomes will be added and sometimes set of chromosomes will be deleted. That is said to be ploidy. And this ploidy, it's of two types. They are euploidy and aneuploidy. So ploidy means what? Sometimes set of chromosomes will be added and sometimes set of chromosomes will be deleted. That is said to be ploidy. And this ploidy, it's of two types. What are they? Euploidy and aneuploidy. So euploidy means the individuals bear more than the usual number of diploid chromosomes. So when you are taking the usual number of diploid chromosomes, that is 2n. So this 2n, it is said to be diploid number of chromosomes. If an individual has 3 haploid sets of chromosome, it is said to be triploid. It is said to be triploid. So you see here, this is the normal diploid chromosomes. With this normal diploid chromosomes, a haploid set of chromosome is added. So here we are getting triploid, or it is also said to be triploidy. Triploid or triploidy. So you see the normal chromosome, that is, what's the normal chromosome? 46. So 46 plus here a haploid set of chromosome is added, that is 23. So here we are getting 69. So 69, that is triploid. When you are taking this triploid, the plants and animals, they are usually sterile. So sterile means what? They cannot give rise to a new individual. So sterile means they cannot give rise to a new individual. And if it has 4 haploid sets of chromosome, it is said to be tetraploid. So now you see here, the normal chromosome is 2n. The normal diploid chromosome is 2n. Plus 2n is added. So we are getting 4n. So 4n. So the normal diploid chromosome, we are adding 2 sets of haploid chromosome. So here we are getting 4n, that is tetraploid. Tetraploid. So see here, What's the normal chromosome? 46. 2n. Diploid chromosome is 46. Plus 2 haploid sets of chromosomes are added. So 23 plus 23, that is 46. So here we are getting 92. So here in tetraploid, the plants with the tetraploidy results in increased fruit and flower size. So see, when there is triploid, the fruits and flowers are sterile. In tetraploid, the fruits and flowers, they are increased in size. Next one is aneuploidy. So in aneuploidy, it's the loss or gain of one or more chromosomes in a set. So aneuploidy means it's the loss or gain of one or more chromosomes in a set. And it's of 3 types. They are monosomy, trisomy, and nullisomy. So aneuploidy, it's of 3 types. They are monosomy, trisomy, and nullisomy. So see here in monosomy, the normal diploid chromosome, so 2n minus 1. Okay, so that is 2n minus 1. 2n means what? 46, isn't it? 46 minus 1. So here there is only 45 chromosome. So in monosomy, there is only 45 chromosomes. Then trisomy. So trisomy means 2n, the normal diploid chromosome. So 2n plus 1. So see here 46 plus 1. So here we are getting 47 chromosomes. So in trisomy, the persons, they possess 47 chromosomes. And nullisomy, 2n minus 2. So 46 minus 2, that is 44. So the monosomy, there is 45 chromosomes, and in trisomy there is 47 chromosomes, and in nullisomy there is 44 chromosomes. Now what's the main difference between euploidy and aneuploidy? So in euploidy, set of chromosomes will be added, and in aneuploidy, one or more chromosome will be added or deleted. That's the main difference between euploidy and aneuploidy. And the next one is Down syndrome. So Down syndrome. This Down syndrome is a genetic disorder and it cannot be cured by medicine. It was first identified by Langdon Down. Langdon Down in the year 1866. In Down syndrome, there is an extra copy of 21st chromosome. So there is an extra copy of 21st chromosome. So here we can say this Down syndrome as trisomy. Why? To the normal chromosome. See, to the normal chromosome, one chromosome is added. That's an extra copy of 21st chromosome, isn't it? So the normal diploid chromosome is 2n. So with the 2n, one chromosome is added. So here we are getting 47 chromosomes, isn't it? So in Down syndrome, there's an extra copy of 21st chromosome. So there it is also said to be trisomy, and here the Down syndrome persons, they possess 47 chromosomes. Coming to the symptoms are mental retardation, then poor growth or poor development, behavioral problems, then weak muscle tone, then the loss of vision, then hearing disability. Last one is gene mutation. So DNA's particular portion is said to be gene. And we know that the DNA is made up of millions of nucleotides. So what are nucleotides? Sugar, phosphate and nitrogenous base. The nitrogenous bases are purines and pyrimidines. So if there occurs changes in the nucleotide sequence of a gene, then it is said to be gene mutation. So define gene mutation. If there occurs changes in the nucleotide sequence of a gene, it is said to be gene mutation. It involves substitution, deletion, insertion, inversion. So when you are taking this DNA, see here there is sugar, then a phosphate group, then here the nitrogenous base. So for example, when here there is guanine. The nitrogenous base guanine is here. This nitrogenous base guanine will be replaced by cytosine. So see, the cytosine will be replaced here. So that is coming under substitution. So this is sugar, then the phosphate, then here the nitrogenous base guanine is here. This guanine, it is replaced by cytosine. That's coming under substitution. And in another one, see here the next one, this is adenine. So here in between these two there is a gap, isn't it? So in that gap, this cytosine is inserted. Cytosine is inserted in between these two gap. So that is called as insertion. And in another one, this guanine, it will be coming here, and this adenine, it will be going here. That is called as inversion. So when you are taking gene mutation, if there occurs changes in the nucleotide sequence of a gene, it is said to be gene mutation, and it is formed of substitution, deletion, insertion and inversion. So coming to mutation, what do you mean by mutation or define mutation? Mutation means sudden change in the genetic material of an individual. It's of two types. What are they? Chromosomal mutation and gene mutation. Then coming to the next one, ploidy. So ploidy, sometimes a set of chromosomes will be added or sometimes set of chromosomes will be deleted. That is said to be ploidy. Ploidy is of two types. What are they? Euploidy and aneuploidy. And this aneuploidy, it is of three types. What are they? Monosomy, trisomy and nullisomy. And the last one is gene mutation. So gene mutation, if there occurs changes in the nucleotide sequence of a gene, it is said to be gene mutation. I hope you all might have understood and you study this mutation as homework.